Hi girls, in this short video we're just going to review the immune responses by the B and T cells. At the top here for each section I've just given you some words and key phrases to remember. So to start off with, we're just going to talk about how the immune response is initially recognised, and that is by macrophages. So macrophages, remembering, are a type of white blood cell that carry out phagocytosis. So they will spot a foreign antigen, and how they do that is by the foreign pathogen with the antigens on it giving off chemical signals. These chemical signals that are detected by the phagocyte are called chemotaxis. It's not essential that you remember that word, but just understanding that pathogens give off signals that alert macrophages is useful. So once we have had the macrophage being alerted to the foreign pathogens present, phagocytosis occurs, and as a result of that, we have an antigen-presenting cell being formed. I'm not going to go into that level of detail again. Hopefully that is part of the course that you are comfortable with. If not, please go back and review your notes. The phagocytic cell, which now presents the foreign antigens on its cell surface, thus becoming the antigen-presenting cell, migrates to the lymphoid tissue. It's in the lymphoid tissue where B and T cell activation occur. Now, the first cell that we come across that starts off B and T cell response is the helper T cell. So, in order of events, it's the antigen presenting cell, migration to the lymphoid tissue, where we meet the helper T cell. Now, the helper T cell, what it does is it detects the antigen-presenting cell and recognizes that it needs to stimulate the humoral response by the B cells and the cell-mediated response by the T cells. And how it does that is by releasing chemicals called cytokines. So let's go on to look at B cell activation. So up here, just to remind you, our key words, we've got B cells, plasma cells, antibodies or free antibodies, and memory cells. So B cell activation starts with something called clonal expansion. Clonal expansion is essentially just rapid mitosis. This B cell that has been stimulated and activated will produce lots and lots of copies of itself. And it does that so that it can then go on to produce plasma cells and memory cells. So if we look at the plasma cells first, plasma cells are a specialized type of B cell that has antibodies, okay? But different from B cells, it doesn't have these antibodies stuck to the cell surface. The B cells will actually release these antibodies, and you can see that in this cartoon as well. So when plasma cells made by B cells secrete free antibodies because they move freely, not attached to a cell, they will inactivate a pathogen by 4 different mechanisms. So they bind to the foreign antigen and either neutralize the pathogen, they will carry out agglutination, they will do antibody binding, or they will induce lysis. So hopefully you are familiar with those 4 different ways that antibodies can inactivate antigens. If not, please go back to PowerPoint number 3 and review the notes from there. In addition to the formation of plasma cells, we also have the formation of memory B cells. So these are stored throughout the body, and their job is to carry out the secondary response. So when we spot the same foreign pathogen again with the same antigenic markers on its surface, The memory cells are stimulated to rapidly produce more plasma cells, which will produce more antibodies at a much faster rate, more rapidly, and a higher concentration, and they persist in the body for longer. Moving on, we're going to be looking at T-cell mediated immunity. So this is when we actually have cells with a specific receptor attached to it. So again, the word specific comes up a lot because antibody receptor sites work like a lock and key. They will only bind to a specific antigen, so thus this killer T cell is not going to work. The T cell response is different from the B cell response because there isn't inactivation. What actually happens is the appropriate T cell will bind to an infected body cell. So this is one of your cells which is infected with viruses, and it will secrete chemicals. And the one that you need to know about is perforin. So perforin is released from the T cell and it punches holes in the cell membrane. So by having these holes in the cell membrane, it induces lysis. This means that our cell is destroyed and therefore the virus loses a host. It can no longer replicate. In addition to the killer T cells destroying the cell, the infected human cell itself will employ a self-destruct mechanism by secreting lymphotoxins. These are another group of chemicals that the infected cell secretes to destroy itself to ensure that the virus doesn't spread. The third thing that I haven't written down but you should also know about is that the killer T cell also secretes lymphokines. So lymphokines, these are chemicals that will induce other macrophages to come to the site of the lysed cell and carry out further phagocytosis at a faster rate. T cells also produce memory cells, and again, these help to stimulate a faster secondary response. But crucially, the difference between T memory cells and B memory cells is that T memory cells remain in the lymphatic tissue. Okay, so they stay in the lymphatic tissue, they do not migrate elsewhere in the body, unlike the B cells. The immunological memory is the same basis. We want to rapidly produce many killer T cells that will act much faster than the initial primary response. Okay, so that's an overview of the humoral and cell-mediated responses. Hopefully that's cleared some things up for you. If you have any further questions, please get in touch.